Hey guys, so I'm Joey. I'm a fourth year that matched into neurology, and I'm here teaching neuroanatomy. So like the anatomy fellows, I've walked you through a lot of the other parts of the body. We're going to walk you through the brain. I'm going to talk about the brain now. We'll do a mid-sagittal, we'll do coronals, horizontals, the brainstem, and the spinal cord. And like all the other videos that the anatomy fellows have made, obviously this is not the final thing. I'm not going to cover the entire practical list, and all of your test questions come from that. So study hard. Okay, so this here is the brain. So first thing when we look at a brain, very basics, you have to be able to identify just the general lobes. So for example, my fingers are resting on what lobe? Good, so that's the temporal lobe. So if we just know our general lobes, we have our temporal lobe here. We'll talk about boundaries in a second. We have our frontal lobe, our parietal lobe, and our occipital lobe. There's two other lobes. So one we can't see from a gross brain. We have to cut the brain in half to see it, and that is the limbic lobe. I'll show you in the mid-sagittal. The other lobe is in this area in here. So first of all, what is this fissure that I'm opening up that my probe is lying in? Good. That is my sylvian or lateral fissure. If we open up my lateral fissure, this entire region in here, if I were to tag anywhere in here, this would be the insula. So the insular lobe is another lobe that has to do with kind of vague combination of senses. So the insular lobe has some taste, some smell, some memory, and some emotional stuff in here. If we look at other lobes, the frontal lobe has most to do with executive function and motor control, and also kind of higher level thinking that makes us human. The temporal lobe has to do with kind of the what idea. So this is where I keep details, I store information, I know what is what. The parietal lobe, which is up here, is the where am I, and kind of a spatial awareness thing. And then finally the occipital lobe is the main visual area. The limbic lobe, which again is in the middle, is mostly for emotion and memory. Okay, so let's talk about some boundaries for these. So my frontal lobe goes all the way back until I hit this large fissure, or sulcus. So this is the primary sulcus. I'll show you how to identify it in a bit, but that is the back of the frontal lobe is the primary sulcus, and then begins the parietal lobe. The temporal lobe is found here, underneath my sylvian fissure. And we know that the temporal lobe stops and changes to the occipital lobe at this landmark here. This is called the preoccipital notch. So again, know some of those basic landmarks of what separates them. There's a few more on the mid-sagittal side, which we'll look at then. Okay, that said, you're never in a practical gonna just get a tag here and say, oh, frontal lobe. You're gonna have to identify specific gyri and sulci. So let's look here. So if we go on the frontal lobe, and I said identify, be specific, you would say superior frontal gyrus. So in this part of the frontal lobe, there's three gyri that are moving anteriorly to posteriorly. So I have my superior frontal gyrus, my middle frontal gyrus, and my inferior frontal gyrus. What if I tagged here on my inferior frontal gyrus and said identify, be specific? So you would tell me that's Broca's area. So on the left side, and the posterior part of my inferior frontal gyrus is Broca's area, which remember has to do with production of speech. So people with Broca's aphasia can't produce speech. This is the famous guy who said tan, tan, tan over and over. Also know that in your superior frontal gyrus up here are your frontal eye fields, which have to do with controlling eye movement. As we get back, I can notice that all three of these gyri will go and eventually turn into a T. So it's going back and then hits a perpendicular landmark, which moves this way. This perpendicular landmark is my precentral gyrus. My precentral gyrus, do you know what its main function is? Good, so that's where my motor cortex is. So if this is my precentral gyrus, then I know that this is my central sulcus, making this my postcentral gyrus. So again, follow back till you hit the T, that's my precentral gyrus, central sulcus, and postcentral gyrus. Okay. A few more landmarks on the front to see. There's another video on cranial nerves, so I won't stress it, but this is my olfactory nerve, or cranial nerve 1. It is separating a couple of gyri. So what is this gyri just medial to my olfactory nerve? Good, so that's gyrus rectus, or the straight gyrus. So you can see it's this little straight gyrus here. Versus laterally, this is my orbital gyrus, which makes sense because my orbit, my eye sits here. And this whole area is called the orbital frontal cortex, which is a kind of vague area that has to do with combining taste and smell for flavor. Next lobe we'll move on to is the temporal lobe. So the temporal lobe, much like the frontal lobe, is looped into 3 gyri. So I have a superior, a middle, and an inferior temporal gyrus. A couple things. Remember, so here's my sylvian fissure, and if I open this up, here's the insula. If I open this up, and hopefully you can see this, so I have my superior temporal gyrus here, and then moving medially from my superior temporal gyrus, basically my probe is pointing in that direction. You probably want to see this on a real brain. But my probe is pointing on the direction of the transverse temporal gyrus, or Heschel's gyrus. This is where my primary auditory cortex lies. So that's known as Brodmann's area 41. That's one of the Brodmann's area that's really important to know. This is where primary auditory is. It's on both sides. So unlike my Broca's, which was here, or my Wernicke's spot, which is here at the back of my superior temporal gyrus, the primary auditory is bilateral. Okay. Identify. Good, so that's the middle cerebral artery, or MCA. So just remember that my MCA lies in my lateral sylvian fissure. We'll look at the circle Willis in a bit and see why it comes out here, versus my ACA is more midline. So that gets to another important part. MCA distribution of vessels, or the vasculature of the MCA, covers about this area of the brain. So if we know our homunculus, which is incredibly important to know, we know that our homunculus out here that's going to be affected by the MCA includes the tongue, the face, and the upper extremity, versus the ACA covers this region in here. And the ACA is going to be covering the trunk and the lower extremity, which is in here. So people with an MCA stroke are going to have a contralateral, because all motor pathways will decussate, all sensory pathways will decussate, a contralateral loss of sensation and motor to the face and to the upper extremity, versus ACA will be contralateral loss of the lower extremity. It's really important to remember all of that. Okay. A couple other things on our temporal lobe while we're down here. Again, we have our inferior temporal gyrus moving out here. But this gyrus here, that goes from the temporal lobe to the occipital lobe, is the occipital temporal gyrus. This is also known as the fusiform gyrus. And this is where, remember I said that temporal lobe has to do with a lot of storing of details? The fusiform gyrus is where we store a lot of these key details. For example, there's a spot in here for facial recognition. So all the faces that I'm familiar with, I know the details of here, and I can see a face and know exactly who it is. So the next thing we'll talk about is the parietal lobe. So remember, the parietal lobe is separated from the frontal lobe by my primary sulcus here. So here's my precentral gyrus, my primary sulcus, making this my postcentral gyrus. Remember, this is the primary sensory cortex. In the same way, this is the primary motor cortex, this is primary sensory cortex. What if I typed here and said, what Brodmann areas does this encompass? Good. So 3, 1, and 2. So the primary sensory cortex is 3, 1, and 2. The primary motor is 4. And this premotor area, which is kind of in here, is 6. There's not a lot of Brodmann areas you need to know, but a couple of them are those guys. A couple other things we can see in the parietal lobe. If I went here to this region, what would you call that? So that's the supramarginal gyrus. I always remember this, it's pretty easy because here, this sylvian fissure makes a margin, and so supra, above the margin, is my supramarginal gyrus. On the other hand, if I go back here, at this angle of my lateral fissure, this is my angular gyrus. So angular gyrus, supramarginal gyrus. Okay. This area up here, identify. So this is just the superior parietal lobule. It doesn't get broken down more specifically, whereas all of this is the inferior parietal lobule. Okay. Again, there's more with these sections we'll see on mid-sagittal, but for now we'll move back to my to my occipital lobe. So the occipital lobe is actually not a lot that you see on the outside. You just need to know that my occipital lobe is all out here. You should be able to identify this area which my probe is lying in. And this is another thing we'll see better on a mid-sagittal, but you should be able to identify it. Good, so that's the calcarine sulcus. So the calcarine sulcus is a sulcus that divides the occipital lobe from inferior and superior visual fields. And if you go right here to the back of the calcarine sulcus, that's where the primary visual cortex lies. That is Brodmann's area 17. That's another important one to know. So primary visual, tip of the calcarine sulcus, Brodmann's area 17. Okay, so now looking from the ventral side, there's a few more things that we can look up on the gross brain. So, first of all, identify this structure right here. So this, actually a lot of you probably won't have on your cadaver, but this is a pituitary gland, so intact pituitary gland. Most of you likely just have the pituitary stalk. So my pituitary stalk is coming out of this region right here. Identify that. So that's my optic chiasm. So I have another video where I'll talk a lot about the cranial nerves, but remember that your optic nerve becomes your optic chiasm, becomes your optic tract, and right in the middle of all of that is my pituitary stalk. This is why any pituitary lesion is going to cause a bitemporal hemianopsia, because I'm affecting my visual pathways. If we move that out of the way, and we go in here, you can see two little mounds in this area. So those right there are my mammillary bodies. And these are specifically important for memory recall. Okay. So we move down our brainstem. So I'm actually going to hold this here. So here's our cortex. And if we move down, and move some of this out of the way, we'll see our circle of Willis, which we'll talk about in a second. But this region at the top, which is mostly covered by my circle of Willis, is going to be my midbrain. And in where my probe is pointing, I show it better on another video. Those are my cerebral peduncles, which is a large part of the midbrain. Then we reach this large bulge here. This bulge is the pons, which is connected to the cerebellum through these cerebellar peduncles. Finally, you have the medulla, which has a couple key landmarks, so identify it right here. So that's my medullary pyramid versus this, which is my olive. For any of these tags, say I tagged right here and I said embryologic origin, you should be able to identify it. So this would come from the metencephalon. So remember that anything in the cortex comes from the telencephalon. Anything in the thalamus or hypothalamic or subthalamic region is diencephalon. The midbrain is mesencephalon. The pons is metencephalon, and the medulla is myelencephalon. I always remember that those last three go in alphabetic order, so that's how I don't get my M's confused. Okay. I'm next going to look at the cerebellum here. So cerebellum is this smaller area, or basically looks like a small brain coming off of the big brain. So a couple things you need to recognize on a cerebellum. One is if I tagged right here and asked you to identify, you would call that the vermis. So the vermis is a large kind of, vermis stands Latin for worm, and it looks like a little worm that snakes between these two large guys. These two guys are my primary hemispheres, or lateral hemispheres of the cerebellum. If I go here, and I ask you what my probe is lying within, you would call that the primary fissure of the cerebellum. This separates the anterior lobe from the posterior lobe of the cerebellum. That's about all you need to know for the cerebellum for this test. Okay, last thing now we're going to look at is my circle of Willis. So this doesn't have all the circle of Willis branches, and in reality, you really should draw the circle of Willis over and over to learn it. But there's enough on here that we can quiz through it. So, identify. Good, so this is my pica, or posterior inferior cerebellar artery. So pica is a large vessel that comes laterally off of this guy, which is my vertebral artery. The other branch that my vertebral artery gives is this right here. Identify that. So that's my anterior spinal artery. So remember, your anterior spinal artery comes off your vertebrals, and then only one of them will move down anteriorly. There's 2 posterior spinal arteries. Okay, our vertebral arteries then combine to make this structure, which is the basilar artery. The first large branch that the basilar artery gives off here is the anterior inferior cerebellar artery, or ICA. So we can see that this pica posterior is moving posteriorly. It would wrap around and cover this part of the cerebellum back here, versus ICA, if this wasn't destroyed, would come and cover the anterior inferior portion. The superior cerebellar artery then is the last branch coming off of my basilar artery. So right here, this large branch, which is moving to cover the anterior portion of the cerebellum, is my superior cerebellar artery. So in the basilar artery system, you have the ICA, you have the superior cerebellar artery, or SCA, and then any other small artery in this area are called pontine arteries. Identify. So this is the posterior cerebral artery, or PCA. So the posterior cerebral artery is the first branch coming off of the basilar artery, and as you can see, it branches in half to become the PCA, which will move posteriorly to go mostly to the occipital lobe. Don't get that confused with this guy. So this is my posterior communicating artery, or PCOM. So PCA versus PCOM. The PCOM connects my posterior cerebral artery with my middle cerebral artery and internal carotid. So this stub right here, the internal carotid you've obviously seen in the neck, you'll see it in the head, but the internal carotid is snaking up here until it reaches the cervical willis. So we'll just see a nubbin at this point. However, the middle cerebral artery, if I pull this apart, you can see that it would, leave the cervical willis, and it's moving laterally. So it's hard to see, but it's going where my probe is pointing here. And that makes sense, because then laterally it shows up in the insula again. The last thing then that we don't see very well in here, so here's my internal carotid, and then we can actually pull it apart. Here we go. So if you look in here, this, after your MCA comes off, the rest, this is my ACA, or anterior cerebral artery. So it is moving forward. And of course, in this region, moving forward. I'll show you that on mid-sagittal. It's going to be terribly hard to see on here, but if there's a small vessel connecting your 2 anterior cerebral arteries, that is your ACOM, or anterior communicating artery. So again, draw your cervical willis, that's the best way to learn it, but you can see a lot of good vessels on here. Okay, and that's all we're going to do for whole brain. We'll move to a mid-sagittal now. Hey guys, so I'm Joey. Now it's just this is so weird. Hey guys, so I'm Joey. I'm a pseudo-pretend kind of neuro fellow. No. So I'm Joey. I'm a 4th year that's going to be helping out with neuroanatomy. So I'm going to be walking you through a couple of videos here. What's going on now?
Welcome to the new fellow. Say hi. Hi, Jerry.
Hey guys, so I'm Joey. I'm a 4th year neuroanatomy fellow, and I'm matching into neurology. So I'll be here to kind of walk you guys through a couple of videos. What?
You already matched. You're going to be a neurologist.